Hi, everyone. This is Professor Beebe, and we are going to go over the second part of the cardiovascular system that focuses on the heart today. So as far as the heart, it is a four-chambered organ, and basically it acts like two pumps that are right next to each other, and we're going to talk a little bit about that. There's a lot of misconception that the heart kind of pumps from one to the next to the next to the next in sequence, when actually the left and right portions are actually working independently, in a sense, the fact that they are filling and pumping at the same time. The heart is comprised of four different chambers. We have the atria, left and right, in the more superior portion of the heart. And then we have the ventricles, left and right, which are the inferior portions and are more the muscular piece of the heart. Those are the pumping chambers, while the atria are just receiving chambers. We do have, you studied cardiac muscle in A&P1, so we know about the structure, we know about the intercalated disc, and we're going to talk about that as far as how that helps us with polarization. The heart is surrounded by a layer called the pericardium, and that's what's actually going to anchor the heart into the mediastinum cavity. So the right side of the pump is going to receive the spent blood from the body. So it's going to receive that blood that's returning via the superior and inferior vena cavas. And it's going to pump it through pulmonary circulation. And pulmonary, we know when we see palm-o or palma, we know we're talking with the lungs. So the right side is going to pump this spent blood, if you will, to the lungs to kind of be revitalized and to dump carbon dioxide and pick up oxygen. And then it's going to return it, rejuvenated, if you will, to the left side of the heart. And then the left side of the heart is going to actually pump it to the systemic circulation. So that's when we're getting this new rejuvenated blood back out to the body systems. As far as the functions of the heart, it generates blood pressure via the heart contractions. We'll talk about that. It routes the blood, whether to the pulmonary or systemic circulations. And once again, those are two separate systems. It has one-way valves that are going to ensure that the blood flows in one way, one direction. Excuse me. And it's going to regulate blood supply by altering the heart rate and force of contractions based on needs. So this is an illustration of the systemic and pulmonary systems, okay? Here, once again, is the right side. This is receiving spent blood from the superior and inferior vena cava. That blood is dumped into the receiving chamber, which is the right atria, and that is then empties into the right ventricle, which pumps it out to the pulmonary system so we can have gaseous exchange in the lungs. Once that occurs, the rejuvenated blood, if you will, gets dumped back into the receiving chamber on the left side, so the left atria, and that's going to flow into the left ventricle, and then we'll have our forceful pump to get it out to the systemic circulatory system. So as far as where it's located, once again, you already know it's in the mediastinal cavity. The apex is the kind of point of the cone, if you will. And the base is the flat part at the superior end of the heart. Sometimes that's a little confusing because when you hear the word base, you think it's going to be at the bottom, when in fact, in the heart, we're talking about the superior end, okay? And the heart, whoops, sorry. And the heart lies obliquely, and you know what obliquely means from A&P1, but it means slightly on a diagonal, with the apex directed to the left, so the 2/3 of the heart mass lie to the left of the midline of the sternum, okay? And when we get to the pulmonary system, that'll help us explain why the left and right lungs don't have the same number of lobes. So this is showing a diagram of the location of the heart in the thorax. As far as the anatomy of the heart, one of the things that I'm sure you've already learned is that almost every organ, gland, structure that we talk about has different coverings, okay, different kind of protective functional layers. The heart is no different. It has the pericardial sac. Once again, that's telling you what it is. Peri means around, and so pericardium means around the heart. So the pericardial sac consists of two layers. There's a fibrous pericardium, which is going to be the tough outer fibrous layer. This kind of acts as an anchor in the mediastinal cavity. And we also have the serous pericardium, which is the thin, transparent inner layer. This is simple squamous epithelium. So it's the thinnest tissue that our body has. And then this will actually be divided into our parietal pericardium and our visceral pericardium. Now, once again, you know these words from A&P1. Whenever you hear parietal, that means of or pertaining to a body cavity wall. Whenever you hear visceral, it means of or pertaining to a vital organ. So the parietal pericardium is going to be that outer layer that lines the cavity wall of the heart. And then the visceral pericardium, which is also called the epicardium, is what actually covers the heart's surface. And very similar to when we talked about skeletal muscle structure in the in A&P1. And remember that space between these two layers, between the parietal and visceral pericardiums, is filled with pericardial fluid. So here is a diagram showing those layers that we just talked about. The heart wall consists of 3 layers. The epicardium is that visceral pericardium. Okay, remember that's the serous membrane, so that's the one that's sitting right on the surface of the heart. The myocardium is going to be that middle layer. This is going to be composed of cardiac muscle cell tissue. And you know that because you hear myo. And whenever you hear myo, you know that has to do with muscles. So don't forget to keep taking these words apart. And then the endocardium, endo is telling you it's the innermost layer. So this is the inner surface of the heart chamber. So this is what's actually lining the cavities within the heart, okay? And then the pectinate muscles are these muscular ridges that we're going to see in the auricles and the right atrial wall. Okay, and then the trabeculae carneae are the muscular ridges we're going to see on the inside of the walls of the ventricles. Okay, so external anatomy, once again, we have 2 atria or receiving chambers, and we have 2 ventricles, which are the pumping chambers. So if we're looking at a cross section of these, the ventricles are much more muscular. They're going to have a thicker muscular wall because they are responsible for pumping, giving that forceful pump to either get it to the pulmonary system or to the systemic. So the blood is going to enter through either the superior vena cava or the inferior vena cava, or that's what's going to dump into the right atria, or it's going to come through the pulmonary veins, okay? And that's what's going to dump into the left atria, okay? And then we have the blood is then going to leave either through the pulmonary trunk, okay, so the right ventricle is going to pump through the pulmonary trunk, and the aorta, and the aorta is going to pump from the left ventricle to the systemic system, okay? So this is an anterior surface view of the heart. This was something you also covered in your lab. And once again, there's a lot of this content that's going to overlap between the lab and the lecture. Okay, and then here is an actual view of this anterior surface of the heart. This is the posterior surface. Okay, and you can see here, this is the superior vena cava. This is the inferior vena cava dumping into the right atria. Obviously, in these images, the blue or red is indicating which blood is oxygenated versus deoxygenated. So this blood that's returning from the systemic system, so through the superior vena cava or the inferior vena cava, are going to be carrying blood that is spent or has used oxygen. Okay, so that is going to be more deoxygenated blood. So that's what's going to be dumping into this, the right atrium, and then on the left side, what's going to be dumping into that is the left pulmonary veins. Okay, now here you can see that these veins are carrying oxygenated blood, because remember that spent blood that's being dumped into the right side of the heart goes to the lungs to become rejuvenated. So when it returns to the heart to get pumped back out to the body, it's actually oxygenated. So this is a good example of where you can't always say that all veins are carrying spent blood and all arteries are carrying oxygenated blood. When we're talking about the left side of the heart, that is not true. It's the exact opposite. All right, so as far as the coronary circulation, the arteries, so the left coronary artery exits the aorta just above where the aorta leaves the heart. So this is where the heart is getting its own fresh supply of blood. So as the left ventricle is pumping out that rejuvenated blood to the systemic system, it's forcing it out into the aorta. It's also then going to pump that fresh oxygenated blood directly into the heart as well. Okay, and that's kind of a great idea if you think about it, because we can't survive without the heart pumping, and so to pump, the heart needs to have that fresh oxygenated blood. So we don't want the heart to have to kind of share blood with the rest of the systemic system. So it has its own circulatory system. Okay, so we have the left coronary artery that exits the aorta. We have the anterior interventricular artery. Okay, this is what's going to supply the anterior portion of the heart. We have the left marginal artery, which is going to supply the lateral wall of the left ventricle. And then we have the circumventricular artery, which is going to supply the posterior wall. And then we have the right coronary artery that exits the aorta, just superior to the point where the aorta exits the heart. And this lies in the sulcus. And remember, a sulcus, just like you learned with the neural system with the brain, the sulcus is that groove, okay? And this is smaller than the left, and this is going to extend to the posterior aspect of the heart. And for that, we have the marginal artery on the right ventricle, and then the posterior interventricular artery, which lies in the posterior interventricular sulcus to supply the posterior and inferior aspects of the heart. Okay, so this is a diagram showing you these arteries within the coronary system. Now, I will say for the lecture exam, I'm not going to be asking you the names of these arteries. That same thing goes with the veins. Remember, the veins will be returning blood to the circulatory system that's already spent. So the great cardiac vein is basically going to drain blood from the left side of the heart. The small cardiac vein drains it from the right margin of the heart, while the coronary sinus is where these veins empty before they dump into the right atrium, okay? Because once again, just like the systemic system, once the blood circulates and services the heart, it has lower oxygen levels and it's spent, so that has to then be dumped back into the systemic system to go to the heart and then to the pulmonary system, okay? As far as the coronary circulation, blood flow is not continuous. Vessels are compressed when the heart contracts, so flow is diminished. As soon as the cardiac muscle goes into diastole, which is relaxation, the flow will resume. So let's talk more about the heart chambers. So like I said before, the atria are basically receiving chambers, okay? We have the right atria, which is going to receive that deoxygenated or spent blood. We have three major openings. So we have the superior vena cava and the inferior vena cava, so receiving blood from the superior portion of the body versus the inferior portion of the body. And then we have the coronary sinus, which once again is returning that spent blood from the heart muscle or the organ to the right atrium. And then we have the left atrium, which receives blood from the pulmonary veins, okay? And then we have the intra-atrial septum. All that is this wall between the two atrial chambers. And remember, the left and right are basically like two pumps that are operating right next to each other. But there's no mixture, or there shouldn't be any mixture or openings between the left and right. Okay. Once again, the intra-atrial septum is the wall between the atria, and there is a remnant of a fetal opening between the atrium. The ventricle, once again, are our pumping chambers. These have a strong muscular presence. The atrioventricular canals are openings between the atria and the respective ventricles. So that's how the blood is going to dump from the right atria to the right ventricle, or the left atria to the left ventricle. The right ventricle opens to the pulmonary trunk to pump that spent blood to the lungs, and the left ventricle opens to the aorta, which is our pathway to the system and the heart. And of course, we have the interventricular septum, which is the wall between the ventricles. So this is a diagram showing you this structure, okay? And I just want to point out, we have these kind of valves here that are going to prevent the backflow of blood. So we'll talk about those too. All right, so the valves, the AV valves, so these are the atrioventricular valves. So they're telling you where they are. They're between the atriums and the ventricles. We know the left and right operate separately, so we know the left atrium is going to dump into the left ventricle, and the right atrium is going to dump into the right ventricle. Each valve has these leaf-like cusps that are attached to these papillary muscles by the chordae tendineae. Okay, the right actually is a tricuspid valve, so it has three of these cusps, and the left is a bicuspid or two-cusp structure, and is also called the mitral valve. Okay, oops, sorry. The semilunar valves then are the valves that are going to pump the blood from the ventricles to either the pulmonary system on the right or the systemic on the left, okay? Each cusp is shaped like a cup, so when the cusps are filled, the valve is closed. When the cusps are empty, the valve is open. And I just want to point out, it's going to be the backflow of blood after that initial pump that's going to back up almost and close those valves. Okay, so this is a diagram showing you. And once again, I just want to, let's see if this is the best image I want to show you. No, this is not the one I'm showing you next. Okay. So once again, these are showing the valves and their function as far as kind of keeping the blood from moving backwards. Okay, and here's those chordae tendineae that are going to anchor these valves. And remember, this is the bicuspid valve. All right, so here's the bicuspid valve. So here is the blood that's dumping from, okay, here we are, dumping from the left atrium to the left ventricle, okay? So here's the left atrium, which just had the blood returned from the pulmonary system through these pulmonary veins, okay? Once that fills, it flows through the bicuspid valve. And then once this pressure increases, that's going to cause this valve to slam shut, And then the contraction of the cardiac muscle is going to cause the blood from this left ventricle to be forced out the aortic semilunar valve into the aorta. Okay. And what's important here is to notice that these are not happening at the same time. These are as a sequential event. Okay. So the atrium is going to fill the ventricle until the pressure increases enough that this valve then closes, the bicuspid valve. And then when we have the contraction of the left ventricle, that's going to force the blood out through the aorta, through the semilunar valve, and then the back pressure will shut these valves, okay? So this is a very helpful diagram, I think, to kind of push you through or to guide you through the blood path in the heart. And you should know the blood path through the heart. So, and we've actually kind of gone through all this, but I'm going to go through it once again, okay? So the deoxygenated blood, remember I keep calling it spent blood, but the deoxygenated blood enters the right atrium. So the right atrium is relaxed. It's accepting this spent blood from the superior vena cava and the inferior cava, and then also from the heart through the coronary sinus. So most of the blood then in the right atrium is going to pass into the right ventricle. And then the right atrium then contracts, and that will push, kind of squeeze the remaining blood into the atrium. And that will complete the right ventricle filling. Contraction of the right ventricle then pushes blood against the tricuspid valve, forcing it to close. And then the closing of the tricuspid valve, okay, so it doesn't flow or so the blood doesn't move back into the right atrium, then actually is going to force the blood against the pulmonary semilunar valve, forcing it to open so that the blood can flow into the pulmonary trunk. Because remember, it has to go to the pulmonary system to get rejuvenated. The pulmonary trunk then branches to form pulmonary arteries, okay, which is going to carry this spent blood to the lungs, where we'll release carbon dioxide and pick up oxygen. The blood then, which is rejuvenated, will return from the lungs via the left atrium through the 4 pulmonary veins. Most of the blood passes from the left atria through the bicuspid valve, okay, into the relaxed left ventricle. Contraction of the left atrium then completes left ventricular filling. So once again, exactly the same process we saw on the right side. And then we have contraction of the left ventricle to push the blood against the bicuspid valve, closing it, preventing blood from moving back to the left atrium. And then the blood will be also pushed against the aortic semilunar valve. And that's what's going to allow the blood to actually enter the aorta. The blood that's leaving the aorta is going to be distributed to the systemic system, so all parts of the body except for the lungs that are supplied by the pulmonary blood vessels. So this, I think, is a really nice diagram that kind of puts it all together without a lot of extra explanation, in the sense that it's a good place for you to kind of go through and make sure you understand the movement of the blood. Okay. So histology. So skeletal muscle, which you studied at great length in A&P1, and cardiac muscle are very different, just like they are very different from smooth muscle. Okay. As far as the skeleton of the heart, we have fibrous connective tissue between the atria and the ventricle. We have fibrous rings around the valves to support them. And then these are also, the skeleton, if you will, is also going to act almost like an insulator. So just like you have insulation on your cords for your lamps or something like that, this heart skeleton is going to provide the same process for the atrium and ventricle. We know that cardiac muscle has those elongated branching cells. They usually contain 1 to 2 centrally located nuclei. We obviously have the actin and myosin filaments. Remember, it is striated, just like skeletal muscle, unlike smooth muscle. We have the sarcoplasmic reticulum, which stores the calcium ions. Okay. But we don't have the cisternae that we saw in the skeletal muscle, so it's a little slightly different. Very, very rich in mitochondria. Okay, but if you think about it, and if you think about the contractility of the cardiac muscle, it needs a lot of energy. So it makes sense that it would be rich in mitochondria. And calcium ions, just like we talked about in cardiac muscle or in skeletal muscle, are going to play a role in this contraction. Okay. We have those intercalated discs. We saw them when we looked at the histology in AMP1. This allows, remember that intercalated disc allows us to withstand a great deal of pressure and stress between the junctions of these cells. Okay. Desmosomes, remember those are those guy wires. We talked about those in AMP1. They are going to hold the cells together. Okay, so these are all part of these intercalated disc. And then the gap junctions, which actually allow the action potentials to move from one cell to the next. Okay, and this allows us to have the atria and the ventricles kind of behaving as one single unit. So this is a diagram showing you these slides. The slide up here probably looks very familiar to one that you had in AMP1, but showing the striation, showing these dark lines, which are the intercalated disc, and then, of course, the centrally located nuclei. So the conducting system. So the heart is an electrical system. We rely on those action potentials to stimulate the heart, to stimulate the cardiac muscle. So we have 2 nodes within the cardiac muscle. We have the SA node, which is also called the sinoatrial node. And this is the pacemaker. So this is the pacemaker of the heart. It's a specialized, these are specialized cardiac muscle cells. They generate a spontaneous action potential. Okay. The AV node is the atrioventricular node. And once again, it's telling you where it is. So this is just medial to the right atrioventricular valve. The action potential is conducted more slowly here than in any other part of the system. And it basically ensures that the ventricles receive signal to contract after the atria have contracted. So after that atria have given their final push to empty its load into the ventricles, then the AV node will be stimulated to give the ventricle signal to contract. The AV bundle passes through the hole in the fibrous skeleton to reach the interventricular septum. We also have right and left bundle branches, and these are going to extend beneath the endocardium to the apex of right and left ventricles. And then we have our Purkinje fibers, which are large diameter cardiac muscle cells with many gap junctions. And remember, the gap junctions allow us to send that message to contract from one cell to the neighboring. And this is what conducts that action potential to the ventricular muscle cells. So here we're going to go through step by step this conducting system of the heart. Okay, so here is the sinoatrial node. Okay, here's the sinoatrial node in the superior portion of the right atrium. Here's the atrioventricular node, or the AV node. Okay, so the heart is at rest, all the chambers are relaxed. So the action potential is generated within the SA node, and this is going to spread from the SA node to adjacent cardiac cells. So that's what these dotted arrows are showing. Then they are going to also conduct the action potential from the SA node to the AV node, to the atrioventricular node. So here's our action potential. It's going to transfer throughout the right atria and then also throughout the left atria. Okay, because remember, these are two pumps activating at the same time. Then these messages are going to go to the atrioventricular node, which is going to innervate, and you can see that here, the ventricles. So the cardiac muscle of the atrial wall is going to contract in response to this action potential. Okay, so that's what you see here, the contraction. Okay, so when the heart beats are under resting conditions, it's approximately 4/100ths of a second is required for action potentials to travel from the SA node to the AV node. Okay, so there's going to be a slight delay between the SA node and the AV node. And that makes sense because we want time for the atria to empty. Okay, so we wouldn't want the atria and the ventricles to be contracting at the same time. It wouldn't make any sense. We need to empty the atria first and then have the ventricles contract. So we need to have that slight delay. Okay. The slow rate of the action potential is due in part to the smaller diameter muscle cells and fewer gap junctions in those intercalated disc. Okay. And then once that message to contract travels down through the septum into the Purkinje fibers, then we have the ventricular myocardium going to be stimulated in the inner walls near the ventricles. So the contraction actually follows this pathway as well. And we're going to see in a minute an image that shows kind of the spiral, not viral, the spiral arrangement of these muscle layers that allow the pump to pump from the apex superior direction. Okay, so it's going to pump from the apex or the pointed area toward the base or the flattened superior order. Right? Like skeletal muscles, cardiac cells have a resting membrane potential and are capable of conducting these action potentials. So the resting membrane potential, just like we talked about in skeletal muscle, depends on the permeability of the plasma membrane. Remember, the plasma membrane's main job is to maintain integrity of the cell. It controls what goes in and what comes out. Okay, so the plasma membrane has a lower permeability to sodium and calcium and a higher permeability to potassium. Remember, the heart is autorhythmic, so it stimulates itself to contract at regular intervals. Remember, that potential comes from the SA node, the sinoatrial node, and once that threshold is reached, the action potential is generated. Okay, so here, this is what we're seeing here. We have that pacemaker potential, okay, leading to depolarization. Then, of course, we always have to repolarize. There's that period where we can have no further contraction, and then we're getting ready for the next phase of the pacemaker. Okay? And you can read through these greater details too. Okay. So the action potentials in cardiac muscle. You're going to notice that this is very, very similar to the skeletal tissue or skeletal process that we saw in AMP1. All right, so we have the depolarization phase. Remember, that's a result in the membrane permeability or the changes in the membrane permeability of sodium, potassium, and calcium. However, membrane permeability to sodium is the primary determinant of this phase. So we have those voltage-gated sodium channels open, bringing about this depolarization phase of the action potential. As those open, sodium diffuses into the cell, because remember, sodium is 10 times higher concentration outside the cell versus inside the cell. So as soon as I open gates, it's going to go from higher concentration to lower concentration, causing that rapid depolarization until the cell is depolarized to approximately 20 millivolts. Okay. And this change it caused by depolarization, excuse me, is also then going to cause changes in the potassium channels. Okay, so we're going to have the movement of potassium is going to be uh, from the inside the cell to outside the cell. Remember, that's going to help us as far as the repolarization later in this process. So early repolarization phase, it's a result, once again, of changes in permeability because we have these charged ions moving across the membrane. That movement of ions causes a charge. And this occurs when those voltage-gated sodium channels, excuse me, and some of the calcium channels uh, close, and then a small number of the potassium channels open. Okay, so this is going to slow movement of sodium. Potassium is 10 times higher inside than outside. So if those potassium channels open, the potassium is going to move out of the cell. And if I have a positive charging moving out of the cell, then the interior face of my membrane is going to become slightly more negative. And that's going to be our repolarization. And then we have the plateau phase. And this occurs as the voltage-gated calcium channels stay open. So the calcium and some sodium move into the cell through these channels. Okay, and this influx of positive ions is going to kind of counteract the initial change by the potassium out of the cell. And then the final repolarization phase after the plateau phase ends occurs when those calcium channels finally close and we open more of those potassium. So if I close those calcium channels, calcium is no longer going to diffuse into the cell, so the cell can reset, and the potassium is going to move along its concentration gradient and diffuse out of the cell. This is going to make the cell membrane slightly more negative and returned it to its resting membrane potential. As far as differences between skeletal and cardiac muscle, in cardiac muscle, the action potentials conduct from cell to cell. Remember, in skeletal muscles, it's conducted along the length of the single fiber. The rate of action potential is slower because of the gap junctions and the smaller diameters of fibers. Versus in skeletal, we have much larger diameter fibers and it's going to be faster. The cardiac muscle has a prolonged period of repolarization called the plateau phase. Okay, remember skeletal muscle resets itself much quicker because it has no plateau. Okay. And this is showing you the comparison of the action potentials between skeletal and cardiac muscle. We have our refractory periods, just like we saw in the skeletal muscle. Absolute refractory period, it doesn't matter how strong the stimuli is, there will be no contraction. And then the relative refractory period, it will, upon added stimulation, we can have a contraction. So this is the ECG. Now, the electrocardiogram, I'm sure many of you have had these or seen them done. Some of you might even be working as patient care partners at Lehigh Valley or St. Luke's, where you actually learn to do a 12-lead EKG. ECG, EKG are the same thing. EKG actually comes from, I think, the Dutch word for the same process. Excuse me, but the ECG is basically a record. It's a print off of all these electrical events that we were just talking about. So you get the six second sheet or printout, and you can measure these different waves to figure out kind of that electrical events and make sure everything's operating and depolarizing and repolarizing how it should be. So this initial wave that you see here is the P wave. And this is that initial depolarization of the atrial myocardium, okay? The QRS complex is this place right here, okay? So this is the ventricular depolarization. And this signals the onset of ventricular contraction. Okay, so this is saying, okay, here we have the message coming from the sinoatrial node. Now it's gotten to the AV node. The T wave, okay, right here, is that repolarization of the ventricles. And then directly after that, we're going to have ventricular relaxation. Excuse me. And then we have the PQ interval, or also sometimes called the PR interval, that is when the atria contracts and begin to relax, the ventricles begin to contract. Okay. And then, of course, the QT, where the ventricles contract and begin to relax. So I'm not going to go through all of these specifics for the ECG waves. Okay. I wouldn't ask you any of this on the lecture test. This is another one of those tables that shows you different types of arrhythmia. So remember, whenever you put A in front of something, it means without. So these are cardiac arrhythmias. So these are conditions that occur because we have lost the typical rhythm. Okay. All right, so the cardiac cycle. A lot of this I think we've already mentioned, but remember the heart is basically two pumps that are working together. It has the right and left, okay? The cardiac cycle is this repetitive pumping process that we've been describing as far as movement of the blood from the right atrium to right ventricle to the pulmonary system, back into the left atrium to the left ventricle, and into the systemic system. Systole and diastole, those are the two measures for blood pressure. Systole is the heart at work, so this is the contraction of the chamber, and diastole is the relaxation of the chamber. Okay, blood moves from areas of higher to lower pressure. So here is the cardiac cycle. So the first step is where the heart is relaxed. So we have passive ventricular filling. Excuse me. So when at the beginning of the cardiac cycle, actually all the chambers are relaxed. At rest, most of the blood movement into the chambers is a passive process. Okay, basically moving from greater pressure to lower pressure. As blood continues to move into the atria, and then into the ventricle for two reasons. Number one, the AV valves are open. Okay, so remember, here's our AV valves. They're open, so the blood can passively move from the atria to the ventricle, from the atria to the ventricle. Okay, and because the pressure is higher. So when the pressure is higher in the atria, it's going to move to the ventricle because it's going to move from higher to lower. Okay. So when this is occurring, this is called passive ventricular filling. And then we have that small burst of atrial contraction, okay? So this is atrial systole. So this is the active ventricle filling. So when we have the contraction of the atrium, excuse me, that's going to push the blood into the ventricle, which up to this point had been a passive process, excuse me. For thinking back to the ECG, this would be the P wave. And then we have ventricular systole, so ventricular contraction. So you see the word isovolumetric contraction. Take the word apart. Iso means same volume. So you have the same volume, or the contraction is happening at the same time, okay? So the action potential passes from that AV node down through the AV bundle, okay, down through the AV bundle, through the bundle branches and into the Purkinje fibers, okay? And this is what's going to stimulate ventricular systole. This is the QRS complex of the ECG. So as those ventricles contract, remember they contract from the apex in a superior direction toward the base, So as this occurs, ventricular pressures increase, causing the blood flow to move toward the atria and close those AV valves, okay? Because remember, we need these valves to close so that when we have full contraction, okay, so we need these to close so that we have full contraction, it's actually going to go to the systemic or the pulmonary systems, okay? And once again, during that time, it's isovolumetric. So the volume has not changed, it's the same. And then we have the second part of ventricular systole, which is actually the period of ejection. Okay, so this is when we can see that contraction is moving up the ventricle, and the ventricular pressure builds high enough that it overcomes the pressure of the semilunar valves. Okay. So here's the aortic semilunar, here is the pulmonary semilunar. So it exceeds that pressure and then can open those valves. As a result, the semilunar valves are pushed open and the blood flows from the ventricles to those arteries. Then we have ventricular diastole. So this is the period of relaxation. So this is the period in the T wave from the ECG. So the ventricles are going to relax. The ventricular pressure decreases. And consequently, the blood, so as the pressure decreases here, okay, in the two ventricles, because this ventricle here is actually pumping out behind this pulmonary trunk, okay? So as the pressure decreases, the pressure, because of the pumping blood into the aorta or out of the semilunar valves, that pressure now has increased. So that is going to cause the pressure to then close these valves. So here's a nice diagram kind of showing this whole process in the cyclical order. There are some great videos in your textbook that actually show, kind of illustrate the pumping, if you will, of the heart. And I think it's very helpful to see kind of how the contraction moves through the ventricular muscle. Okay. At rest, contraction of the atrium is not necessary for heart function. However, during exercise, we need this process to happen a little bit quicker so that we can have a greater portion of the blood, 300 to 400% more blood than during rest. Ventricular systole. This is actually just showing you or kind of giving you more details about the whole process that we've already talked about. But the ventricular muscles start to contract. Remember, systole means contraction at work. This causes those AV valves to close, which is the beginning of ventricular systole. And when we're looking at measurements, okay, we're going to talk a little bit in a minute about volume. There's about 120 to 130 milliliters of blood in the ventricles left from last contraction or last relaxation. So this is referred to as the end diastolic volume. And then the period of, and I'm never going to ask you the pressures in the right ventricle versus left ventricle. I think that's a little bit beyond the scope of this class. But in your lab, we do talk about end systolic volume. So just like end diastolic volume, the EDV, was the volume at the end of that relaxation stage, and systolic volume then is the period at the end of ejection, end of contraction. Okay. Once this occurs, ventricular pressure is going to fall very rapidly, which makes that pulmonary trunk aortic pressure higher. Okay. And this is going to cause blood to flow back toward the relaxed ventricles and cause those semilunar valves to close. Okay. And then we're going to go back to the beginning where we have the passive ventricular filling. Okay, so our ventricles are in the relaxed state, and we're going to have that passive filling. And this accounts for about 70% of all ventricular filling. This diagram I think is nice because it kind of puts everything that we've just been talking about, it lines it all up so you can see them all happening together. As far as the lub-dub, okay, the heart sounds, the first sound that you hear is the lub, okay, and that's basically the closing or the slamming of those AV valves. And then the second sound that you hear, the dub in lub-dub, is actually the closure of the aortic and pulmonary semilunar valves, okay? And this is, remember, occurs at the beginning of that relaxation phase of the ventricles. We can have some abnormal heart sounds, such as murmurs, when we have damage to the valves. So if we for some reason we don't have a completely closing valve, we might have some swishing or gurgling sounds because the valve is not creating that tight closure. Okay. This is showing you where the valves are in the thorax so that you can hear that lub-dub that we've been talking about. Okay. All right. So I'm just going to talk briefly about arterial blood pressure, mean arterial blood pressure. Um, it's slightly less than average blood pressure in the aorta. It's basically a equation that calculates the cardiac output, so that's the amount of blood pumped by the heart per minute, and you multiply that times the resistance, okay? So the peripheral resistance, so whatever is resisting the movement of blood. So whether that's that your arteries are less elastic, so the blood is kind of hitting a harder wall, so it's not passing on the force, it's decreasing the force. Or if you have clogged arteries or buildup on the walls of the arteries, this all causes resistance. All right. Here is a diagram showing you some things that can impact mean arterial pressure. So heart output changes to meet the demands of the body. Okay, so we have intrinsic and extrinsic regulation. Intrinsic regulation is basically normal heart functioning, and extrinsic involves the neurohormonal control. So for intrinsic regulation, a lot of that preload has to do with the amount of stretch of the ventricular walls. The greater the stretch, the greater the force of contraction. This is also called Starling's law. Okay. And then we also have afterload. So that's the pressure that the artery or the ventricles must produce to overcome the pressure of the aorta and move the blood into the aorta. Okay. And then we have extrinsic regulation. We talked about the neural system in AMP1, and we talked about the parasympathetic versus the sympathetic nervous systems. Remember, the parasympathetic system was the one that was kind of in control or in charge during times of relaxation. In the heart, this is going to be supplied by the vagus nerve. So this is going to decrease heart rate, and it's going to hyperpolarize the heart. And then we have sympathetic stimulation. Remember, sympathetic is in the times of stress or higher demand. So this is supplied by the cardiac nerves. Okay. And these are going to increase the heart rate and the force of contraction. And then we have hormonal control, such as epinephrine and norepinephrine from the adrenal medulla. And this is going to occur in response to emotional excitement, also stress, and increased activity. Okay. So this actually takes longer to respond. However, it actually lasts longer than the direct sympathetic stimulation. The heart and homeostasis. We have barioceptors that are monitoring blood pressure in the walls of the heart. This is, as we know from AMP1, remember the medulla oblongata plays the role in cardioregulatory, and it can then, once it receives the information from the barioceptors, regulate and increase or decrease heart rate. We also have chemoreceptors, and chemoreceptors we talked about in AMP1. We talked about the neural system and different types of receptors. Chemoreceptors are going to be embedded within the vessels, and they're going to be they're like little litmus tests, if you will. They're going to measure pH, they're going to measure carbon dioxide, they're going to measure oxygen levels in the aorta. So if we have prolonged lower oxygen levels, this is going to cause an increased heart rate, because if we have low oxygen levels, then we need the circulatory system to speed up so that we can increase the heart rate, so that we can have that exchange or have that rejuvenating effect for the blood happen faster. Okay, and you can go through this in more detail. I'm never going to ask you for the specific name of the nerves, okay? So don't worry about that, okay? And lastly, obviously, if we have an increase in extracellular potassium, that's going to decrease heart rate, because we need that resting membrane potential. So if we have an increase in extracellular potassium, we're not going to have that differential we need to reach our action potential. So if that happens, we have the heart rate and the stroke volume will decrease. And if we have a loss of action potential conduction, that's called a heart block, right? A decrease in extracellular potassium causes hyperpolarization. So if we hyperpolarize, so we kind of go lower than our resting membrane potential, so we actually have to have a greater stimuli to depolarize. So it takes slightly longer to reach that depolarization phase. Okay. If we have an increase in extracellular calcium, remember that's going to allow us to have a greater force of contraction. And then if we have a decrease in extracellular calcium, that's going to increase heart rate and can lead to tetanus or tetany of the skeletal muscles. Heart rate increases when blood temperature increases, and heart rate decreases when blood temperature decreases. You can read through these effects of aging on the heart. Many of these are, I'm sure, familiar to you. Okay, here's another table kind of giving us the summary of all of these. I'm not going to be asking you questions about stenosis or patent ductus arteriosus or any of these. Okay. Myocardial infarction, you can read through these and kind of see, I think this is a great diagram that kind of shows how interdependent all the systems are. And that's it for chapter 20. So please, if you have any questions, let me know. And I will talk to you all soon. Thank you.